So I've tried to update the presentation to include those with our new analysis, but to be honest, I still don't think we've got a good statistical analysis for our frost data. So we'll go through it and hopefully you can see some of the major differences. So frost and chilling, and that's what I'm going to be talking about, and understanding the different factors that influence that. I work for the Department of Agriculture in WA. This is not just a WA project, we also have collaborators. Michael Laws, who works for the University of Adelaide, and he runs our trials in Loxton in South Australia as part of this national project, and Troy Fredericks, who works in Toowoomba with Didi, and he runs the program, the trials up there. So I've just got a bit of a question for you guys because I'm not really familiar with your frosting environment that well. So if you can all put your hands up and then drop them down as we go down, about what temperatures you expect to start to see frost damage occur at in this environment. So yeah, look, you're already onto it. So these we'll say are screen temperatures, for example. So at 2 degrees, do you guys expect to see frost damage? Yep, okay. 0, -2, -4, a lot. This is just a photo of my son. It's not accidentally in there. He's 1 year old. That was when we were irrigating my trial last year and he was dribbling almost as much to irrigate it. So, but I mean, the next question I've got for you is how long do you think it'll be before we start to see frost tolerant varieties? Do you reckon it's going to be when James starts school in about 2015? When James is in high school in 2020? When he turns 1 in 2030? Or when he leaves home? My guess is it's probably going to be sometime after he leaves home if we get there. So a bit of background on frost research for those of you who don't know. Traditionally there's been limited research into breeding for frost tolerance. In wheat, a lot of the initial work was done in the 1960s to the 1980s by Bill Single, based out of Queensland. And then in 2002, Troy Fredericks started working on frost in that northern environment again, and has continued working on that up to now. In barley, most of the work didn't start till about early 2000s, and the late 1990s, going on to that. And basically, there's been reproductive frost tolerance reported in barley from some of this work, which has come out of that South Australian environment. So basically, the University of Adelaide have developed what they are describing as frost tolerance barley, and they've done that by breeding with Japanese material. And this has led to what they've described as a 2H and a 5H source of frost tolerance. This material consistently suffered less frost damage at flowering when they ran their experiments in the southern environment in South Australia at Loxton. However, when Troy screened it in Queensland in 2004 to 2007, he didn't find it any better than any of the commercial varieties that they were already growing up there and running in their trials. This is the Queensland environment, probably similar to here, when you get your mild winter frosts. This is the trials that Troy runs, and you can see with his early planting, early times of sowing, he gets very devastating frosts and a lot of freezing damage and a lot of tissue damage from those frosts. I mean, that looks like it's been sprayed. We would never see this type of damage in Western Australia in a commercial crop. Do you guys see that sort of damage here? No. Yeah. So, I mean, that's a very extreme environment in Queensland, in those trial sites that they're running there. So that's when GRDC decided to fund this project. That's the background that GRDC decided to fund this project with. And really what we're trying to do is characterise the frost environment nationally. So we're taking detailed meteorological measurements to try and determine the similarities and differences between different frost events, between different seasons, and in the at sites in South Australia, Queensland and Western Australia. And we're also doing detailed scoring of frost damage just at flowering to try and determine a level of damage for a particular frost event. And we measure that by looking at frost-induced sterility, which I'll describe how we go as we go forward. So there's 3 trial sites. Mine's at Keeballing in Western Australia. Michael manages the one in Loxton in South Australia, and Troy does the one in Toowoomba in Queensland. We've got 10 entries in the trial, 3 wheats, 7 barleys. The barleys include Japanese material with this 2H and 5H source, which are reputed to vary in that southern environment, and we just want to compare the rankings of these lines between events within each environment and then also between the seasons and across sites, to see if we can get some consistencies. This is just to give you an idea of where the trial sites are. So Keeballing is basically about 2 hours inland from Perth, and this is the number of days with frost in September, so pretty low, only 2, compared to where we are here in Young. You know, you've got between 10 and 15 days in September when you get frosts. Loxton is also less than 2, and Kingsthorpe is probably between 5 and 10 days of frost in September. So Keeballing, this is my Keeballing trial site from last season. It's basically about a 350mm growing season environment, and generally the farmers on average produce about 2.5 tonne a hectare. Good years without a frost, they will go over 3 tonne. When they get hit, they're back down to a tonne. So it's pretty much a big impact on their cropping production in that environment. This is Michael's trials in Loxton in South Australia, 250mm growing season range rule. Commercial crops there around 1.5 tonne. So we are working in different production environments as well, especially when you go and compare that to the Queensland work, which most of their trials are based around a Kingsthorpe and Hermitage, where they've got a lot of soil moisture, and the commercial crops are up at that 4 to 6 tonne level. So we used numerous times of sowing from early April to get our material flowering from July through to October. So we've got a heap of material which we can actually look at the frost damage on. We use similar measurements, layout measurements across the different sites, just different frosts. So this is my 2009 trial at Kibelling. You can see obviously I have to go out and irrigate quite extensively to get decent establishment because it doesn't rain out there very early. Initially we picked basically 5 times of sowing from early April through to early June, and this is basically the range in what commercial growers will grow. In this environment, no one will really try and sow wheat before the 12th of May or before the second week in May because of the risk of frost. But as we've done this, we've worked out that we actually need to put in more times of sowing, and we actually need them spaced more evenly at the start to try and make sure that we've always got material flowering across that window. So during a frost event, we actually have a weather station which will send us a text message and tell us that there's been a frost event. And so that goes off when the air temperature gets to 2 degrees. So we then go to the trials and tag all the material which is just about to flower. We do dissections to stage every different line for the lime yellow anther stage, and then we go and tag all the heads that are morphologically at that stage in that same plot. And then we come back 3 to 4 weeks later during mid-grain field and collect the heads and score the frost damage. So these are the methods that we're using across all of the sites to try and develop our data, to collect the data. So the tagging methods, you can imagine, are quite labour intensive. With the barleys, this is basically where the lime green anther stage are. Most of them are basically at full boot stage or Z45. With the wheat, it's pretty much traditional flowering when you get those first couple of florets coming out of the middle of the head. We try and tag a bit earlier than that, or the head's just before that. So yeah, here's a picture of what the barley generally looks like at the development stage we're tagging at, and the wheat obviously with full head emergence just prior to flowering. The sterility is basically just a count of how many grains have formed. So you go and count how many grains have actually formed, how many are sterile, and calculate a sterility index.
You just look at the first florets here.
Say that again? Main stems? So
first florets or secondary florets in wheat rather than barley?
So no, in wheat we look at the first and secondaries, yeah. Wheat's a lot harder to score, obviously, and that's why we've actually gone to leaving it longer, so we get reasonable grain size to make it easier to actually physically feel the grains without having to pull the whole head to bits. So, but yeah, it's very labour intensive. So this is just some of my results. This trial's been running since 2008, but our 2008 results we've pretty much all discarded because we were worried about inconsistencies between our scoring methods and just getting some decent hold of the data. So this is the data from Western Australia from 2009. These are the minimum temperatures, and you guys are already onto what screen temperatures and plant temperatures. You'll obviously understand the difference between that, so I'll skip over that when I come to that in a minute. But all the temperatures I'll present are actually the canopy temperatures, that canopy height where the heads were during that frost event. So you can see, we don't really have very cold events in WA. I mean, minus 2.2, that was quite cold that year. And this is the range of sterility we got in the wheats and the barleys, 10 to 30%, 10 to 20%, whereas in the wheat, under the same frosting conditions, we're getting up around 100%. Then these later few events, we're getting less levels of damage in the wheat. So graphically, this is basically presenting the data for you to actually give you an understanding of the variability we've got in the data. So these are just the observed means plus or minus the standard errors for those particular varieties. It hasn't been analysed or transformed or anything. But you can see, I mean, there's a couple of points of what you take home from this graph. The barleys, for the same range of frost events, have a lot lower levels of sterility compared to the wheats. Everyone already knew that. Barley's more frost tolerant than wheat. I'm probably not telling you anything there. Under some conditions, we actually do pick up differences within the barley. So under this frost event on the 26th of September, you know, these Japanese sources of barley and sterling had a lower level of frost damage than Gilbert. And the same thing we observed for wheat. Young, unless the frost event, had a lower level of sterility compared to the wild catcham and the kite. But, you know, there's a huge range in the sterility levels that we do observe. And that's proving a big nightmare in this project, trying to work out how we can reduce the variation, the within-plot variation within our data. But under some conditions, you know, there are varietal differences. But, you know, that's one event out of, I think there's only 9 plotted there, where we've actually managed to find some actual varietal differences. So barley, a lot more tolerant than wheat, but there wasn't really enough damage to discriminate between the lines. Wheat, last event, wild catcham, you know, was more tolerant than kite and young, but more damage than kite and young. South Australia had similar data, and I'll show you that in a minute. But, you know, it's only one event out of 10 that actually gave us these differences. So is it real or not under most frosting conditions? Can you make agronomically useful decisions? Yeah, I mean, it's questionable. This is the South Australian data for 2008, 2009 and 2010. And I'll put this in because it gives you an understanding of how much variation we're attributing to the actual varieties and how much variation we're looking at within plots. So you can see here, this is just for the wheat data set. You can see that in 2008, you know, the most variation we ever attributed to variety was in 2009, where we got up to 64% of the variation due to genotype. But there's still a heck of a lot of variation within the plots. So there's most of the time, most of the variation is within the plot, not between the varieties. Despite that, yep.
Do we know whether that's due to temperature variations between one plot and bits and pieces? Or?
The biggest problem we've got is we're counting individual heads. And as anyone who's looked at a frosted crop will know, some heads get completely wiped out and some heads get no damage. And so you've got a huge binomial distribution in your data sets, and a lot of that variation goes to the plot level. And that's what we're trying to work out, how we can analyse that in the best approach.
I'm actually surprised that you're getting that much genetic effects.
So, well, some years we are, yeah. And South Australia is a very good climate. And if you come, yep. If you come back to 2010, you can consistently rank young, kite and wild catcham to low, mid and high sterilities. And in 2010, I think pretty much, I don't think the error bars are on this one, but it's in your paper. These events are significantly different between varieties. And that's what we've tended to find. If we can get a damage level which gives us somewhere in this order of, you know, 10 to 50% damage, then we can pick up varietal differences. If we get more damage than that, we don't have any varietal effects. If we get less damage than that, there's no varietal effects. So under some conditions you can, but these are, and I'll go on to try and explain how we're trying to work out what climatic factors are driving that. So all of the sites also have a weather station, and it's not just every plain old weather station, it's also got net radiation sensors to try and measure the cold radiation coming in from outer space at night and the warm radiation coming up from the soil and from within the crop canopy. We're also measuring soil moisture and the heat balance, leaf wetness sensors to try and work out when the actual dew formation on the leaves actually freezes, and obviously the canopy temperatures at different head heights. I can skip over this because most of you know that when you measure screen temperature on a frost night, it's actually shielded because there's no air movement through the screen, and so you don't end up with a good estimate of the air. So most of all the temperature data I'll present is of unshielded temperature probes.
Did you just want to make a comment on your temperature differences between screen, top canopy and lower canopy?
There's no correlation. Depends on the frost event, but generally, yeah, your screen temperatures are higher than what the plant's actually experiencing. But we haven't been able to find a decent correlation between screen temperature and what the plants experience. We know it's different, but it's very difficult to look at screen temperatures and get an understanding of what your plants are experiencing. And that's because of the disconnect, that insulation effect. And it depends on how much air movement you get. Like if you get a little bit of movement, you know, if this temperature that you're recording inside in a shield will actually be okay. But sometimes it's not. So this is just, I've just plotted here the hours where we got less than 4 degrees, less than 2 degrees, less than 0, less than -1 and less than -2. And this is for 2009 at my Kiballing trial site. You can see and these are basically the range of sterility we've got in the wheat. So with these events, we got 100% sterility. With these later events, that's when we got the 5 to 50% sterility, and that was in the last one when we got pretty much nothing. So we got this data, and then we went, oh, well, what would happen if, how does this compare to the Loxton site? So when I plot the Loxton site on there, you can see initially that Loxton, it does get lower, it does go down to -2 as well with the plant canopy temperatures, but the frequency that it gets down, less than 0, those yellow bars, is a heck of a lot less. And so that environment is a lot less severe than the West Australian screening environment in which we're looking at. So their frequency of frost, even though we get down to probably a similar intensity, the number of frosts they have in a year is a lot less. And that's probably one of the reasons why they can actually pick up these varietal differences. So after that, we decided that we would try and actually tag on a schedule instead of after a frost event, just tag every week and look at the sterility after that to try and see if we can find some control levels where we actually got reasonable grain set in these gaps where we didn't have any frost. So that's what we did last year. You can see this is my temperature data from last year. I just got clobbered with frost because we had a very dry finish to my season. I had lots of frost. I tagged every week, but as you're going to expect, I got a heck of a lot of sterility. So this is the 2009 data. My minimum temperatures, I got a lot colder this year as well. -5, that was a -5.1, that was a new record. And we got reasonable levels of damage in barley. 50 to 80% damage on this variety, and that was significant. We had significant varietal differences. 2nd of September, we also picked up some significant varietal differences, and later on this 14th of September. I'm not really going to go into the barley in this presentation, but feel free, if you want information on the barleys, then please feel free to come and talk to me afterwards. But the wheat's pretty much 100% sterility until we got to these last few events when we started to actually get some more discriminating events. This event here, the event 12 on the 24th of September, I actually had to irrigate that day to stop drought stress becoming a big impact on my trial. We had a frost that night and it just smashed everything. So we ended up with 100% sterility, which is a bit annoying because that was experimental stuff up which really mucked up my results. But anyhow, these things happen. What we can pick out, we're also looking at different hour thresholds for a 10-day period around flowering. And you can see the main thing that's different between these later events where we are getting those lower levels of damage is that there's a lot less frequency of temperature less than -1. So basically there's not a lot of period here where the temperature was less than 0, which really makes us question about what sort of temperatures do the plants actually need before they will get damaged. This is a plot of the sterility for the 3 weeks across time, and really all I want you to make an observation about is this data compared to this data. This is that event 12 where I ended up with 100% sterility because I irrigated. So, and this is over time, but basically what you can see here, we actually tagged an extra time this year. This is the flowering data, and this is the young microspore data, which is when the wheat's at booting stage. And you can see here, there does appear to be more varietal differences between the wheats under some conditions. See this time, at this event, kite had lower levels of sterility than wildcatcham and young, whereas later on, it was the other way around. Young had less sterility than kite and wildcat